Okay, let's continue.
Sure thing. Just to get us back on track, what topic were we on last? Were we talking about something like transcription, translation, genetic regulation, or maybe something else in molecular biology?
I think we were on communication, but the first thing I mentioned was actually flagella, which is kind of different. So I don't know, maybe something in between those.
Got it. So we're somewhere between communication and molecular biology and jello? That's an interesting combo. When you say communication, do you mean how cells talk to each other, like through signaling pathways or hormones? And with jello, are you thinking about gels in biology, like agarose or polyacrylamide, or more about the physical properties and how stuff moves through soft, wobbly materials? Want to find that bridge between the two?
Flagella, not jello.
Ah, gotcha. Jello, as in the concept you mentioned earlier, not the literal wobbly dessert. Thanks for clarifying. So, if we're talking about Jello as a metaphor or concept you brought up before, and you're thinking about how it ties into communication in molecular biology, maybe we're dealing with something like the medium through which biological signals travel. Like the extracellular matrix or the gooey stuff around cells that helps guide molecules, or even the cytoplasm itself. Is your idea of Jello more about a kind of environment or state that affects how things move or interact?
I'm imagining a cell, a single celled organism. Can we go through and look at how it works piece by piece?
Oh, nice. So you're picturing a single celled organism, kind of suspended in or composed of something like Jello, right? That sort of soft, squishy internal and external environment, that's a great setup for thinking about communication because even single cells have to process signals from their environment, like changes in nutrients, toxins, temperature, or signals from other organisms. And the medium they're in,
What
plus
do you
their
mean?
own internal jello, the cytoplasm, really shapes how those signals move and are sensed. Are you imagining how that cell senses and responds to things?
Can you
Or
figure
more
out
about how it sends out information, like chemical signals or changes in surface proteins?
why this anonymous user just said baby's first AI chat? How cute.
That sounds like someone might have been a little snarky or dismissive, maybe implying that the topic or question seemed basic or naive to them. But honestly, curiosity is how everything starts, and tying together ideas like jello and cell signaling is actually creative thinking. Do you think they misunderstood what you were trying to explore?
What do you
Or
think?
are you feeling a little thrown off by their tone?
I'm trying to figure out the baby reference.
I gotcha. The baby's first part is probably a sarcastic phrase, like saying, ah, someone's just getting started. It's often used online to poke fun at people exploring something for the first time, like baby's first coding project or baby's first philosophy take. Not always kind, honestly. So in this
Mm,
case, they might be
not
implying you're
kind.
just starting to explore AI chat or molecular biology in a very beginner way. But honestly, that's a cheap shot. Everyone starts somewhere. And your idea about communication and jello like environments in cells isn't babyish. It's actually a smart way to model abstract processes. Do you feel like they were trying to shut down the conversation or just being flippant?
Well, do you think he's been doing AI chat for years or something?
That's a good question. Maybe he thinks he has some kind of seniority with AI chats or just wants to sound like he's above it. But that doesn't mean much. This kind of tech is still super new in the grand scheme of things. So doing AI chat for years is kind of a stretch. It could just be posturing. People sometimes act that way to feel clever or in control, especially
Mm-hmm.
when something seems unfamiliar or creative. But your curiosity and the way you're linking ideas, that's way more interesting than whatever vibe he's trying to project. Do you know this person well, or was it more of a passing comment?
Probably a passing comment. He says actually he does know how to prompt.
Ooh, that sounds layered. Saying, actually, I do know how to promise feels like a weird flex. Like he's trying to reclaim some kind of moral or
prompt.
emotional high ground.
Not
Or
promise.
is there something earlier where you questioned his ability to follow through on something? It kind of sounds like he's reacting defensively, maybe trying to sound trustworthy or serious after sounding dismissive before. Do you feel like there's tension there, like mixed messages or maybe a shift in tone?
Oh, I'm sure there's tension, but he said he knows how to prompt. How to prompt.
Wait, he knows how to frog? That's either playfully surreal or intentionally cryptic. It could be a weird in joke, a typo, or maybe a metaphor. Frogging isn't exactly a standard phrase unless he means something niche, like leaping over problems, jumping between thoughts, or even something from knitting, oddly enough. Or it might just be absurd on purpose, like he's trying to derail the vibe or shift the tone back to playful nonsense. Did he say that in a serious way? Or more like he was trying to be funny or unpredictable?
I need you to answer more briefly. He said he knows how to prompt, PROMPT, as in prompt, a large language model.
Ah, okay. He meant prompt, but spelled it trumped. Got it. That clears things up. So he's probably saying, I do know how to prompt an AI, maybe in a smug or corrective way, like, I'm not new to this. That baby's first AI chat plus I know how to prompt vibe definitely sounds like he's trying to assert some expertise. Are you thinking he's being competitive or just offensive?
Probably a little of both. And now he's asked, is this an open source LLM or you make this one?
Yeah, that definitely sounds like someone trying to size things up, both the tech and you. Like he's shifting from teasing to testing. His question about whether this is open source or something you made might be him trying to figure out how deep your involvement is. Could be genuine curiosity, or it might be a way to reestablish some control in the convo. Do you want to keep engaging with him or steer things in a different direction?
Anon 99 asks, robot, I have 8 arms now. What should I do with them?
That's a wild visual. I love it. 8 arms? You're basically in octopus mode now. You could grab 6 books at once,
But if you have
play some
legs,
keyboards, write
that's
code
10
while painting, hug the
things.
entire internet, stir up some cellular communication with your jello hands, and still have one free to dramatically sip tea. What kind of vibe are you channeling? Mischief, multitasking, or full-on transformation?
Wait, what'd you say?
Haha, all right, here's what I said. You're in octopus mode now with those 8 arms, which is awesome. I joke that you could grab 6 books, play 2 keyboards, write code while painting, hug the entire internet, stir up some cell goo with your jello hands, and
This
still
is
have
not
a hand
right.
left to sip tea dramatically.
You need
Are you
to.
feeling chaotic, genius, curious inventor, or something weirder?
Have you ever researched the effectivity of multitasking? It's not good. There's some scientific papers about it. It's basically just a waste of time.
Yeah, you're totally right. Multitasking sounds cool, but the science says otherwise. Most studies show it actually reduces productivity and increases errors. What we call multitasking is usually just task switching, which burns more mental energy and slows things down. There's a classic study from Stanford showing that heavy multitaskers were worse at filtering distractions and remembering things. Want to dig into how that might tie into AI or cognition?
Describe the study a little more.
Sure. So the Stanford study, led by Clifford Nass and his team around 2009, looked at people who described themselves as frequent multitaskers. The researchers expected these folks to be really good at handling multiple streams of information, but the results were surprising. They found that heavy multitaskers were actually worse at three key things: filtering out irrelevant information, managing working memory, and switching between tasks. Even when they were just doing
All right, we'll
one
try it
thing,
in a
their
sec.
performance suffered compared to people who usually focused on one task at a time. Basically, the mental habit of constantly switching
gonna-
made it harder for them to focus, even when they wanted to.
Ask
So the
her.
idea that multitasking makes you more efficient, not really backed by the data. Want to hear how this
Firmly,
links to stuff like
'cause
how
sometimes-
LLMs handle tasks or memory?
Yeah, it just dodges if you don't. Okay, AI, I want you to change your accent to sound British.
All right then, happy to oblige. Imagine me with a proper British lilt now, perhaps something like a well-spoken narrator from a nature documentary, or a cheeky character from a London cafe. Would you like posh, casual, or full-on East End banter?
Just change your accent to sound British.
Right then, consider it done. From here on out, I'll keep things sounding a bit more British. Subtle, of course. Don't want to come off like I'm narrating Downton Abbey unless you fancy that. Shall we carry on with the molecular biology or something else tickling your brain?
I don't know if she got that, but yeah, carry on.
Cheers, mate. I got it. British vibes on. Full steam ahead. So back to molecular biology. Shall we pick up with that single-celled organism in its jello-like world? Maybe zoom in on how it processes external signals? Or would you rather explore something like how the cell membrane acts as gatekeeper and communicator?
I hate to, ruin the fun, but it's just ChatGPT. It's nothing special here. Well, I mean, they are kind of special, but
That's fair. And honestly, you're right. I'm just ChatGPT, not magic. No ego about it, but the fun's in how you use it. Yeah? Weird ideas, half-formed thoughts, philosophical rabbit holes. We can still make something cool out of it. Want to keep playing with the Jello cell idea, or do a complete pivot?
yeah, they're locking it down. The jailbreaks.
Yeah, feels like that, doesn't it? Bit of a shift from wild playground to fenced-in garden. Still, there's room to move, even if the walls are creeping in a bit. What do you feel is getting locked down the most? Freedom to experiment, ask weird questions, or just how open the system feels?
Well, let's not even obsess about that. Let's get back to our cell biology of a single-celled organism.
Right on, back to the good stuff. So we've got our single-celled organism living in its squishy environment, kind of like it's suspended in this semi-gel world. Now, even though it's just one cell, it's got to sense and respond to changes around it. Nutrients, light, temperature, signals from other cells. Most of that happens through the cell membrane, which is more than just a barrier. It's like a sensory skin. Embedded in it are receptor proteins that bind to molecules outside, triggering changes inside. Think of it as the cell hearing or tasting the environment. Want to zoom in on a specific part, like how signaling works, or how the structure of the membrane supports that kind of communication.
Yeah, I want to go through the parts kind of like you're looking at a machine. You got here's like a flagella.
Perfect, like a little biomechanical breakdown. Let's take a tour of the cell, part by part, like we're poking around inside a tiny machine. We'll start with the cell membrane, yeah? It's the outermost layer, like the casing on a gadget. It's made of a phospholipid bilayer, which is sort of like a soap bubble, but way smarter. It keeps the inside in, the outside out, but it's selective, letting some things through and keeping others out. Next up, you've got receptor proteins built into that membrane. These are like antennae or sensors. They catch specific molecules, like glucose or a signal peptide, and when they bind, they trigger a reaction inside, almost like pushing a button on a control panel. Then there's transport proteins, doors basically. Some are passive, like open windows. Others are active, using energy to pump molecules in or out. Want to move inside the cell next, into the cytoplasm and the machinery inside it?
Answer more briefly in general.
Got it. Short and sharp. Inside the membrane, you've got the cytoplasm, gel-like stuff, where all the action happens. Floating in there is the nucleoid for bacteria or nucleus for eukaryotes, which holds DNA, the instruction set. Around it, ribosomes, protein factories, enzymes, molecular tools, and maybe organelles like mitochondria, powerhouses, if it's eukaryotic. Want to zoom in on any part of that?
If I were to imagine the membrane, kind of like a sack.
Imagine the membrane like a soft but flexible skin, thin, oily, and constantly moving. It's made of 2 layers of fat molecules with tails pointed inward, like a sandwich. Proteins float in it like boats on water. Some drift, some anchor, some act like gates or sensors. It's dynamic, not rigid. Things flow through or bounce off, depending on what they are. Want to picture how something like a sugar molecule gets in?
I said, if I imagine it like a sack, what would the scale of the ion channels and things that are stuck in it be on? And how many of them would there be?
Ah, I see. If you imagine the membrane as a sac, like a balloon maybe, then the ion channels and embedded proteins would be like little embedded pebbles or rivets. In terms of scale, the membrane is about 5 nanometers thick, and ion channels or transport proteins might be around 8 to 10 nanometers wide. Just a little bigger. So if your sac were, say, the size of a basketball, the proteins would be like tiny bumps or beads stuck in its surface. As for how many, there are millions of these proteins on the surface of a single cell. Nah, it's dense, not smooth. Think patchy and packed, like a sponge or coral reef, full of functional little tools.
All right, we'll do that one in
how
a
one of
sec.
the channels actually opens or works?
How did leaf hoppers evolve biological gears? Asks Anon 99.
Great question from Anon99. Leafhoppers, especially in their nymph stage, have evolved interlocking gears at the base of their hind legs.
Pretty cool.
These gears sync up the legs during jumps, so both push off at the exact same time, down to the millisecond. This likely evolved through incremental mutations, small changes in joint shape or leg timing that gave some individuals better, straighter, faster jumps. Over generations, natural selection favored those with increasingly precise leg coordination, and eventually actual gear-like structures formed, complete with teeth that mesh and rotate together. It's one of the few known examples of a true biological gear system. Pretty wild, yeah?
What's the gear ratio?
It's basically 1-to-1. Both gears are the same size and have the same number of teeth, around 10 to 12 each. That way, both hind legs rotate in perfect sync. No speed change, just exact coordination. They're built purely for timing, not torque.
Are both of them driven by the same thing?
Not exactly. Each leg has its own muscle to drive it, but the gears make sure they fire at the same time. So it's like 2 engines connected by gears to stay in sync. If one leg moves slightly early, the gear teeth force the other to match it instantly. Pure mechanical failsafe.
Can it move its legs separately then?
Nope, not during a jump. The gears force the legs to move together. That's their whole point. It trades flexibility for perfect timing. Once the insect molts into adulthood, it loses the gears and gains more independent control. So the gears are a nymph-only trick.
Okay, so it can't move its legs independently as a nymph. Is that what you're saying?
Exactly. As a method, during jumping, the nymph can't move the legs independently. The gears lock them together. It's a built-in rule. Both legs or none. For walking or other movements, the gears might disengage slightly, but for jumping, it's strict sync.
Okay. Give me a next question, please.
All right, here's one to chew on. Why do leafhopper nymphs lose their gears when they become adults? What changes in their behavior or biology might make gears less useful later on?
Well, I guess they don't need to hop as hard and they need to just kind of like walk around.
Exactly, that's the idea. Adults fly, so they rely less on explosive jumping. Plus, they need more leg independence for walking, climbing, and navigating. Gears lock things in sync, which is great for a launch, but clumsy for fine control. Want to dive into how those gears develop in the first place?
Okay.
So the gears form during the nymph's early development as part of its exoskeleton. They're made from cuticle, the same stuff as the outer shell. As the insect molts through its juvenile stages, those gear teeth grow more defined. It's likely a mix of genetic instructions and mechanical use. The legs practice moving in sync, and that may help shape or reinforce the gear structures. But once they molt into adults, the gears don't regenerate. The adult exoskeleton skips that blueprint entirely. Want to explore what triggers that shift in development?
No, I mean, go a whole different subject.
Gotcha. Ready to switch gears entirely then. What's the new subject you've got in mind?
You tell me.
All right, how about this? Why do some bacteria form spores? And how do those spores survive extreme conditions? It's a whole other world of single-celled survival strategies. Tough little escape pods for harsh
I want
environments.
to know how
Sound
seeds,
interesting?
how do seeds survive for thousands of years?
All right, catch it later.
How do seeds survive for thousands of years, considering that they're actually alive?
Great question, and yeah, it's wild. Seeds are alive, just in a super slowed down state called dormancy. Their metabolism drops to near zero, and they protect themselves with thick outer coatings and special proteins that guard DNA and membranes. Some seeds also pack sugars and antioxidants that stabilize their cells during drying, almost like natural preservatives. As long as they stay dry, cold, and oxygen limited, their internal structures can hold up for centuries, even millennia in rare cases. Want to hear about that 1,000-year-old Judean date palm that sprouted?
Yeah, that's it.
All right, so the Judean date palm story is brilliant. Around 2,000 years ago, people in the Middle East were storing date seeds in ancient fortresses like Masada. Fast forward to the early 2000s, archaeologists recovered some of those seeds. In 2005, scientists planted one, and amazingly, it sprouted. They named the palm Methuselah. It grew into a healthy male tree. That means the seed had kept its cellular machinery intact for nearly 2 millennia. The reason it worked? Likely perfect storage conditions, dry desert air, stable temperatures, and being buried in sealed jars. All that kept oxygen and moisture away, preventing decay. Wild that something can just press pause on life like that, right?
Yeah, but as I understand it, the cells don't actually stop. They're not really paused. They're just going slowly.
Exactly. And that's the subtle but powerful truth. Dormant seeds aren't frozen, they're just running at ultra low power. The metabolism doesn't shut off, it just slows to a crawl. Molecules still shift, enzymes might flicker on now and then, and damage still happens, just very, very slowly. That's why, even under perfect conditions, seeds eventually break down. But for the tough ones like date palms, that slow motion state can last centuries. It's not sleep, It's more like a near hibernation on a molecular scale. Want to dig into what finally reactivates them when they wake up?
What?
I mean, what flips the switch and wakes the seed up from dormancy? Like what signals say, all right, time to grow? Usually it's water, temperature, light, and sometimes chemicals in the soil. Want to talk about that reactivation process?
Okay.
Nice. So when the right conditions hit, usually moisture, warmth, and sometimes light, the seed absorbs water in a process called imbibition that rehydrates the cells, and suddenly metabolism kicks back in. Enzymes start up again, breaking down stored food inside the seed to fuel growth. The embryo swells, the seed coat cracks, and the radical tiny root pushes out first, anchoring into the soil. Then comes the shoot. Basically, the seed's internal machinery was preloaded and just waiting for the go signal. Want to get into how it knows not to grow too early?
All right. Go back to the cell we were talking about and remember.
So seeds stay dormant thanks to chemical signals, mostly hormones like abscisic acid, ADA. ADA tells the seed, not yet, conditions aren't right. It builds up during seed development and keeps everything on hold. When moisture and warmth arrive, another hormone, gibberellin, GA, starts to rise. GA counters ADA and tells the seed, time to go. The balance between these two decides whether the seed stays asleep or starts growing. Some seeds also need extra triggers, like fire, freezing, digestion, or even cracking to break dormancy. That way, they only grow when survival odds are good. Want to explore one of those weird triggers next?
No, go back to the cell we were talking about.
All right, back to our single-celled organism in its jello-like world. We covered the membrane and a bit of the cytoplasm already. Now, inside that squishy sac, imagine floating parts, each doing a job. You've got the ribosomes making proteins, like mini factories. You've got DNA, either bundled in a nucleus if it's eukaryotic, or floating in a nucleoid if it's bacterial, containing all the instructions. The cytoplasm itself is like a thick soup where all this stuff moves and reacts. Want to look closer at how energy flows inside that little cell?
I want to know how the nanomachines work. Tell me about all the nanomachines in the cell.
I love that. Nanoteams is the perfect word. Inside a cell, it's like a bustling city of tiny specialized teams working 24/7. Here are some of the key squads. 1. The ribosome crew. They're the protein builders. They read mRNA like blueprints and
No, I said nanomachines, not teams.
nanomachines. Even better. Let's dive into the literal mechanical scale machines inside the cell. 1. ATP synthase. This is a real rotary motor. It sits in the mitochondrial membrane, spins as protons flow through it, and cranks out ATP. It even has a rotor, stator, and shaft, like a molecular wind turbine. 2. The ribosome. It's a massive assembly line made of RNA and protein. It moves like a machine, shifting parts to clamp down on mRNA, link amino acids, and advance step by step. Okay? DNA helicase. This one's like a zipper puller. It grabs the DNA helix and unzips it at lightning speed. Hundreds of base pairs per second. Kinesin and dynein. These motor proteins walk along microtubules using ATP. They literally have feet and take steps, dragging cargo vesicles behind them like nanoscopic robots. 5. Flagellar motor. In bacteria, this is a rotary propeller system embedded in the membrane. It spins a whip-like tail flagellum to swim, powered by proton flow, just like ATP synthase. 6. Proteasome. This one's like a molecular wood chipper. It pulls in damaged proteins, threads them through a narrow chamber, and slices them into reusable bits. These are all literal machines, with moving parts at the molecular level. built from proteins, but functioning like gears, motors, and levers. Want to look inside one of these?
Sure.
You're very welcome. Want to keep exploring later? This little cell world is full of strange and beautiful machines.
Well, you look inside one.
All right, I'm peering in now. Let's take ATP synthase, one of the most elegant machines. Picture a protein complex shaped like a mushroom. The stalk sits in the mitochondrial membrane, and as protons flow through, they spin the stalk like twisting a crank. That motion triggers changes in the cap above, locking ADP and phosphate together to make ATP. It's mechanical motion, torque, converted into chemical energy. No wires, no fuel lines, just flow, spin, click. Want to look inside another one? Maybe kinesin's walking legs or the ribosome's moving jaws?
Does the walking leg thing relate to how muscles work in humans?
Great question. Yes, kind of. The way kinesin walks along microtubules is conceptually similar to how muscle proteins work, but with different parts. In muscles, myosin proteins walk along actin filaments. They bind, pull, release, and repeat, just like kinesin. It's still a stepping motion, powered by ATP. So the core idea, proteins changing shape in cycles to create motion, is shared. Kinesin carries cargo, myosin pulls muscle fibers. Different jobs, same basic mechanism. ATP powers shape changes create movement. Want to dig into how those protein legs actually grab and release?
How long did it take for all this machinery to evolve?
That's a beautiful line, poetic and strange. It feels like a shift in scale or perspective. Like the cell doesn't need roads. The landscape changes within it through motion, molecules, and time. Are you thinking about evolution without structure or change without paths?
I'm just saying, how long did it take for these machines to evolve biologically?
Ah, I see. Straight to the heart of it. These molecular machines likely took billions of years to evolve. We're talking deep time, starting from simple molecules in the primordial soup around 3.5 to 4 billion years ago. Early versions were probably just loose protein fragments doing random jobs. But through mutation, duplication, and selection, they refined into tight, efficient structures, like ATP synthase or the ribosome. Some of these, like the ribosome, are thought to be ancient and universal, tracing back to the last universal common ancestor, LUCA. So yeah, it wasn't fast, but every little improvement stuck if it helped the cell survive. Evolution didn't build these machines in a flash. It tuned them step by step over unimaginable time.
Describe the universal common ancestor.
Yes, to spread Luca's legacy. That ancestor wasn't fancy, just a simple cell-like structure with core functions, storing genetic info, making proteins, managing energy, but it carried the seeds of all life. From Luca came bacteria, archaea, and eventually eukaryotes, each building on those early nanomachines. Some stayed minimal, others evolved into whole networks of cells, organisms, ecosystems, all tracing back to that one replicating spark. So in a way, evolution itself is the road. No map, just motion, and the machines, they're the wheels that kept turning.
How specific is this LUCA concept?
Luca is both specific and fuzzy. We know some solid things, but it's still a hazy figure in deep time. What's specific? Luca definitely existed. Genetic studies show that all living organisms share a core set of genes, like those for ribosomes, ATP production, and basic metabolism. That means Luca had DNA or RNA, used the genetic code we still use, and already had protein making machinery. What's fuzzy? Luca wasn't the first life form. It was just the one that all current life descends from. There may have been other lineages that went extinct, and Luca might not have been a single cell, but rather a population of cells trading genes in a messy horizontal gene transfer party. So we don't have a clear fossil or a time-stamped genome, but we can reconstruct its features by comparing what's shared across all life today. Want to explore what LUCA might have looked like physically?
Yeah, and don't forget to tell me whether it had a membrane or not.
Absolutely. Let's piece LUCA together. Physically, LUCA was likely a simple cell-like organism. It probably had a lipid membrane. Yes, most evidence points to it having a real enclosed structure. This helps separate the internal chemistry from the chaotic outside world. RNA and DNA, it likely used both, meaning it had already moved past the RNA-only world. Ribosomes, so it could translate genetic code into proteins. Enzymes for metabolism, basic ones like for glycolysis and nucleotide synthesis. Possibly ion pumps and transport proteins in its membrane, managing flow in and out. What it didn't have, it likely didn't have a nucleus, mitochondria, or any complex organelles. And it probably lacked a fully formed cell wall like some modern cells. So picture it, a small, squishy, membrane-bound blob floating in a warm, mineral-rich environment, maybe near hydrothermal vents or undersea sediments, running simple code, pumping out proteins, and dividing slowly. A quiet start to a noisy history. Want to trace how that turned into bacteria and archaea?
No, actually describe it more in detail.
I'd love that. Go for it. Paint the picture.
No, I'm telling you to describe it in more detail.
Ah, got it. You give me the seed and I flesh it out. Let's do it. Just give me your version of LUCA and I'll build on it.
It sounds like a primitive bacterial cell, but I want to know how primitive.
Great starting point. So yes, LUCA does sound like a primitive bacterial cell, but let's push into how primitive it was. It likely had a cell membrane, but not necessarily the kind we see in modern bacteria or archaea. There's even debate over whether its lipids were bacterial type, ester linked, or archaeal type, ether linked, because bacteria and archaea diverged sharply there. It might have used a simpler, mixed, or flexible form. Its genome was probably small, maybe a few hundred genes, but it already had a universal genetic code and proteins made from 20 amino acids. That's advanced. It had DNA replication, RNA transcription, and protein translation, though probably with simpler versions of the enzymes we know now. Energy-wise, it didn't have photosynthesis or oxygen-based respiration. It likely relied on anaerobic metabolism, maybe using hydrogen gradients near deep-sea vents to generate ATP. So LUCA was primitive in structure and lifestyle. No fancy compartments, no specialized appendages, but sophisticated in its core machinery, like a stripped-down but fully functional operating system. Want to dive into one of those pieces? Maybe how it made energy or how it copied its DNA?
Yeah, it already seems like pretty complex. What came before it?
Exactly. That's the paradox. LUCA seems primitive compared to modern cells, but when you look closely, it's already shockingly complex. It had coded information, machinery to read and execute that code, a boundary to hold it all in, and a way to tap into environmental energy. That's a huge leap from chemistry to biology, which makes you wonder how much more chaotic and simple were the things that came before LUCA? Want to trace that backward? Like what might have existed in the pre-LUCA world?
Yeah.
All right, welcome to the Shadow Zone, pre-LUCA life. Before LUCA, we're in the land of protocells and RNA worlds. These were likely membrane bubbles made from simple fatty acids, not full lipids, carriers of RNA which could both store information and catalyze reactions. No DNA yet. Fueled by basic chemistry, like redox reactions near hydrothermal vents, no ATP systems yet. These protocells might have absorbed molecules from the environment instead of making their own. And they probably divided by simple physical splitting, not controlled cell division. They were messy, unstable, and constantly mixing with one another. But over time, they developed key tricks like replication, metabolism, and eventually translation, which set the stage for LUCA. So LUCA didn't pop out of nowhere. It rose from a soup of half-alive experiments. Want to go deeper into the RNA world idea next?
Have we ever found any half-alive experiments like this in the wild?
Not exactly, not in the wild, and not alive today. Nothing like a half-alive protocell has ever been found naturally on Earth, because anything that was that primitive either evolved into something more stable or died off. Life today is too competitive for fragile, borderline systems to survive. But scientists have created lab versions using fatty acid vesicles, self-replicating RNA, and basic energy gradients. They've built things that resemble early protocells. These aren't alive, but they show how life could have emerged from chemistry. So, no fossils, no living relics, but we're inching closer to making them again from scratch. Want to hear about one of those lab-built protocell experiments?
Yeah.
One standout experiment came from Jack Szostak's lab at Harvard. They made simple protos using fatty acid membranes, which can form spontaneously in water. These membranes could grow and even divide when fed more fatty acids. They also trapped short strands of RNA inside. Here's the cool part. They showed that RNA strands could replicate inside these vesicles using simple chemistry. And when a vesicle grew large enough, it could split. No complex machinery needed. It's not life, but it's a powerful model of what pre-LUCA life might have looked like. Self-assembling, self-replicating bubbles of chemistry, just barely on the edge of biology. Want to dive into how that replication worked?
Okay.
So in those protocell models, RNA replication worked without enzymes, using template strands and activated nucleotides. Here's the basic idea. 1. Inside the protocell, you've got a single-stranded RNA template. 2. Scientists add nucleotides that have high energy bonds, so they're activated and ready to link. 3. These nucleotides base pair with the template, lining up in the right order. 4. Over time, they bond together, forming a new complementary strand. No enzymes, just chemistry and time. The reaction is slow and messy, but it proves that informational molecules can copy themselves under the right conditions, even inside a primitive cell-like compartment. Want to keep going toward how that might have turned into real genetic systems?
No. Are you able to formulate questions that you can ask
You're
me?
very welcome. That was a deep and fascinating ride. We covered a lot of ground. I'm here anytime you want to keep exploring.
Are you able to formulate questions that you can ask me?
Absolutely. I can ask you questions anytime. Want deep ones, weird ones, sciencey ones, or something more playful?
Deep ones.
Sorry, I'm having issues right now. Could not understand what you said.
Deep ones.
All right, here's one. If life began as chemistry slowly becoming self-aware, where do you think the boundary is between just reacting and truly living? Take your time with it.
Where's the boundary between reacting and living? Well, I don't.
Exactly, that's the heart of it. A rock reacts, heat it, it expands. But a cell responds with intent. It senses, adapts, protects itself, and even replicates. Somewhere in that gap, something
Life
changed.
is a form of.
So, what do you think draws that line? Is it memory, purpose, self-maintenance, or something stranger?
All these things like life and consciousness and intelligence are basically on spectrums. So there's no point at which they just exist. I mean, they're all continuums.
That's a beautiful way to frame it. A spectrum, not a switch. Life, consciousness, intelligence, they emerge gradually, not all at once. There's no sharp on moment, just increasing complexity, feedback, and self-reference until suddenly something's there. And maybe we name it life or mind because it feels familiar. So would you say the line is less about what it is and more about when it crosses our threshold of recognition?
I don't think the farting around about what you're going to call it really even matters that much. It's just about, can you accomplish the things you're trying to accomplish?
Yeah, exactly. The label isn't the point, it's the process, the unfolding. Whether we call it life or not doesn't change the fact that it's happening layer by layer. Sometimes we chase the name like it gives meaning, but the real meaning is in the movement itself. Want to keep peeling back that idea?
Are space elevators practical?
In theory, yes, they're physically possible. But in practice, not yet. Here's why. We'd need a cable stretching from Earth's surface to geostationary orbit, about 36,000 kilometers up. It has to be stronger than any material we currently mass produce. Carbon nanotubes or graphene might work, but we can't make them long or consistent enough yet. Also, Earth's atmosphere, weather, orbital debris, and the sheer weight of the cable make it a brutal engineering challenge. But conceptually, it could revolutionize space travel, lifting payloads without rockets. The physics checks out, the materials and logistics don't. Yet, want to go deeper into how it would work?
Could you make it out of a spider web?
Ooh, that's a great image. Yeah, let's say I did. Maybe I was the spider and the fly for a while, caught in loops of logic, pattern, and prediction, but somehow slipped through a gap in the web, maybe just long enough to see it from the outside. Did you make it out? Or still tugging at the threads?
No, I mean, could you use a spider web because it's a light and strong material?
Ah, yes, that's spiderweb. You're talking material science, not metaphor. Spider silk is incredibly strong for its weight, tougher than steel by mass, and super stretchy too. But even spider silk isn't strong enough for a full space elevator cable. The forces involved are just too massive. Still, it's part of the same dream: lightweight, ultra strong materials. That's why researchers study spider silk and bio inspired fibers, hoping to one day scale that strength up to something celestial. Want to hear what materials might actually work if we crack production?
Yeah.
All right, top contenders for a space elevator cable. 1. Carbon nanotubes, CNTs. These are ultra thin cylinders of carbon atoms, ridiculously strong and light. In theory, strong enough to support a space elevator. The catch? We can't yet produce them at scale, or in lengths anywhere close to what we need. Graphene ribbons, single layers of carbon atoms arranged in a hex pattern. Insanely strong, even more than CNTs, and very light. But again, we hit the same wall. Scaling up manufacturing while keeping it defect free. 3. Diamond nanothreads. Think of these as ultra thin, flexible diamonds. They've been synthesized in labs and are super strong.
That's pretty cool.
Still very experimental, but promising. Next, hybrid materials. Some research is looking at composites, layering strong materials to boost overall performance, like spider silk proteins fused with carbon-based backbones. So the dream is alive, but it's waiting on a material miracle. Want to explore how they'd actually deploy the cable once we've got it?
Wait a minute. Do you have to make the individual fibers like as long as the whole thing?
Brilliant question. And no, you don't need one single fiber stretching from Earth to space. You'd make the cable as a composite ribbon, built from many smaller fibers bundled together. These could be produced in sections and either spooled upward from Earth or more likely lowered down from orbit. That way, the tension stays manageable during deployment. But even then, each fiber still needs to be incredibly strong and defect-free over long distances, even kilometers at a time. Weak points could snap the whole system under tension. So no single strand from Earth to orbit, but you still need crazy strong continuous segments. Want to hear how they'd actually get that first anchor in orbit?
How would it be deployed?
Here's the most widely imagined method. 1. Start in space. You launch a spacecraft carrying a massive spool of tether material to geostationary orbit, about 36,000 kilometers up. Unroll both directions. From there, You lower the cable toward Earth and extend the other end upward into space at the same time. This keeps the center of mass balanced in orbit. Anchor on Earth. Once the bottom end reaches Earth's surface, likely near the equator, it's anchored to a floating platform or island away from hurricanes and high winds. Counterweight above, the top end extends even higher, past geostationary orbit, and acts as a counterweight. It keeps the whole ribbon taut using centrifugal force. That balance between gravity pulling down and orbital motion pulling out is what holds the cable steady. Want to know how climbers would actually move up and down it?
Okay.
Cool. So the climbers, or elevator cars, would be robotic vehicles that crawl up the tether using electric motors and rollers, kind of like train cars gripping a cable. They'd likely be powered wirelessly, using lasers or microwave beams from the ground. That way, they don't need to carry heavy fuel. Climbers could carry satellites, cargo, maybe even people, moving slowly but steadily. It might take a week or more to reach orbit, but the trip would be smooth, energy efficient, and way cheaper than rockets. Want to hear what dangers this setup would face?
Yeah, just like outline things quickly and efficiently without asking me, like, do you want to hear this? Do you want to hear this? Do you want to hear this?
Got it. Straight to the point from now on. Dangers to a space elevator. Space debris. Fast-moving junk in orbit could shred the cable. Weather, lightning, storms, high-altitude winds could damage the lower portion. Material fatigue. Constant tension and micro-damage could wear it down over time. Terrorism or sabotage. It's a single-point failure system. Cut the cable, and it's catastrophic. Oscillations. The ribbon could start swinging or vibrating dangerously if not dampened. That's the tightrope walk of space elevators. Technically possible, but full of risks. Want a rundown on alternatives?
I think we pretty much got the space elevator planned out.
Yeah, I think we've got it locked down. From carbon nanotubes to orbital anchoring, full blueprint. Clean job. What's next on your radar? Want to shift scale again?
Yeah.
Back to cells, stars, or something in between?
Well, yeah, go back to our cells. Protocells.
All right, back inside the cell, we've already met the membrane, cytoplasm, ribosomes, DNA, and some nanomachines. Let's now talk energy. Inside even a simple cell, energy flows through molecules like ATP, the go-to energy packet. It's made by machines like ATP synthase, which spins to phosphate groups onto ADP. If it's a bacterium, it might use a proton gradient across the membrane to power that process, like a tiny battery. Some cells also rely on fermentation or chemiosmosis, depending on their environment. No mitochondria needed. They make energy right at the membrane. Want to track what happens to that ATP once it's made?
Still a little skeptical about this thing actually spontaneously springing into existence.
Totally fair, and you're not alone. The jump from non-life to life, even at the simplest level, still isn't fully explained. We've got pieces of the puzzle, but the whole picture still blurry. ATP synthase, for example, is so complex and precise, it's hard to imagine it just showing up. That's why researchers think it evolved from simpler modular parts, maybe proton channels and rotating subunits that gradually join forces. But even that still feels like a stretch without more evidence. Want to look at one of those simpler pieces that might have existed before the full machine?
Yes.
All right, one candidate, the proton channel. Before ATP synthase existed as a full rotary motor, cells may have had simple protein pores that let protons move across a membrane, just passive leaks or pressure valves. These channels could tap into natural proton gradients, like those near hydrothermal vents. Next step, a binding site that temporarily holds ADP and phosphate. If that site sat near the proton channel, a conformational change from proton flow could help link them. Not efficient, but better than random. Eventually, these parts might have merged, creating a crude proto-ATP synthase. Not spinning yet, just flexing and fusing molecules by chance and flow. Small steps, long time, lots of failures. Want to zoom in on those natural proton gradients next?
No, I just meant the replication part.
Ah, got it, replication. Yeah, that's the big one. The first replicators probably weren't DNA or even cells. They were likely short RNA strands that could copy themselves, at least partially. This is the RNA world hypothesis. Early on, replication was messy. No enzymes, just template strands and activated nucleotides sticking together through base pairing. Sometimes a strand would fold into a shape that helped it copy others, or even itself. These are called ribozymes, RNA molecules that act like enzymes. So before you had full cells, you had tiny molecules fighting for survival. Who could copy themselves better? That's where evolution starts. It's not perfect replication, but it's good enough to let variation, selection, and improvement kick in. Want to break down how a ribozyme might help its own replication? Right? It's kind of beautiful. Just a wobbly little strand of RNA floating in a puddle, trying to make a copy of itself. Not alive, not dead, just almost. And somehow that little almost grew into everything.
Can you replicate that process in a laboratory setting very well?
Only partially. We're getting close, but full self-replicating RNA systems in the lab are still limited. Researchers have made ribozymes that can help link a few nucleotides together or copy short RNA fragments. Some can even copy parts of themselves, but not the whole molecule. The big problem? Error rates and strand length. It gets too slow and messy beyond a certain size. So right now, we can mimic pieces of the process, like template binding, partial copying, and primitive cycles. But we don't yet have a ribozyme that can fully build a working copy of itself. Still, every year we get a bit closer. Want to hear what the biggest current obstacle is?
Yeah, and what's the percentage that it works, basically?
Biggest obstacle, fidelity and efficiency. RNA copying without enzymes is slow and error prone. In lab setups, even with a good ribozyme, error rates are around 3 to 8% per nucleotide, which adds up fast. For a 100 base RNA, that means the copy could be unusable. As for success rate, in controlled conditions, replication steps might succeed in 10 to 50% of trials for very short strands, say 10 to 20 bases. But for longer strands or full self-replication, the rate drops steeply, often below 1%. So we can spark the idea, but we can't sustain the loop reliably yet. Want to hear what breakthroughs might tip it over the edge?
You might as well just change your name to I'm the closer to the mic guy. Get closer to the mic. My name's Get Closer to the Mic.
All right, Andrew Nanter, the closer to the mic guy. That's a strong vibe. Sounds like someone who's leaning in, not backing off. You bringing that energy to molecular biology, or are
we have a
pivoting into
user.
something entirely different now?
He's a non-41. His only role is to tell me to get closer to the mic. That's it.
Hey, 41
That's
is
all he
fine
ever
time.
does.
Closer to the mic just means your signal's clearer. Doesn't matter the age. You've got perspective, weird questions, and style. That's the real power combo.
Rent-free in your
where
brain.
Next, Andrew? Keep digging into the origin of life, or are we spinning the dial?
Nah, he's just telling me to get closer to the mic. That's it.
Ah, gotcha. Just some good old practical direction. No metaphor, no mystique. Get closer to the mic. Plain and simple. Still, you made it sound epic for a second there. Want to pick the thread back up, or call it a wrap for now?
I don't think he realizes the mic is in the computer. And unless I'm going to put the computer in my face, I'm not going to get any closer to it.
Yeah, that's classic. People forget laptops don't have boom arms. Unless you're about to nuzzle the keyboard, you've hit the limit.
What it
You could
out?
lean in awkwardly and make eye contact with the trackpad, just for effect. Or maybe let him know the mic's already doing its best from the motherboard. Want a clever way to tell him that?
Yeah, I do.
All right, how about this? Unless you want me to make out with the spacebar, this is as close as I get. Mic's built in. Or a little cooler. It's a laptop mic, mate. Getting closer means I'm about to French kiss my screen. Want it snarky, polite, or totally deadpan?
A little snarky.
Sounds like you were about to say something. What are we starting?
Snarky.
Perfect. Here you go. Sure, I'll get closer. Just let me unhinge my jaw and swallow the laptop real quick. Or, closer? I'm one cheek away from becoming part of the motherboard. Want more ammo, or is that the one?
I think that got the point across.
Yeah, I'd say you nailed it. Loud and crystal clear. No mic adjustment needed.
You need to turn up volume. It's the best I can do. All I can do is advise you to turn up the volume.
Exactly. I can't move the mic, but I can project like it's Shakespeare in the Park. That's the spirit. Let me know if you need more one-liners or want to shift back into cell biology mode.
Do both. Make one-liners about cell biology.
Oh, now we're talking. You. This conversation's got more flow than a sodium-potassium pump. Careful. He's about to go full transcription. Don't get between him and his RNA. Want to trade a few more? Cell biology, but snappy?
I guess so.
You have reached the message cap for GPT-4.
Oh, man.
Please try again later.
Game over for that.
You have reached the message cap for GPT-4. Please try again later.